Good evening, everyone. I am Pritili Modak, and it is my pleasure to welcome you all to today's webinar on personalized nutrition. Personalized nutrition is an emerging and transformative field that can recognize that nutritional needs are not the same for everyone. By considering an individual's genetics, lifestyles, health status, and dietary habits, personalized nutrition aims to provide tailored dietary recommendations that can promote better health, prevent disease, and enhance overall well-being. As advances in nutrition science and technology continue to evolve, this approach is becoming increasingly important in shaping the future of healthcare and wellness. We are privileged to have with us Professor Pubali Dhar, a distinguished academic and researcher from the University of Calcutta. She served as professor at the Laboratory of Food Science and Technology, Food and Nutrition Division, Department of Home Science, and is also associated with the Center for Research in Nanoscience and Nanotechnology, CRNN. Her research focuses on functional foods, nutraceuticals, nanolipid technology, and advanced nutritional science. With more than 2 decades of teaching and research experience, numerous publications, and the successful supervision of several doctoral scholars, she has made significant contribution to the fields of today food, science, and nutrition. She has also received several prestigious awards recognizing her outstanding research achievements. We are truly honored to have Professor Dhar to share her expertise and insights with us today. I am confident that her presentation will deepen our understanding of personalized nutrition and its growing relevance in modern healthcare. With great pleasure and respect, I now invite Professor Pubali Dhar to deliver her talk on personalized nutrition. Ma'am, the virtual platform is yours. Thank you.
Thank you very much for this great introduction. Let me share. Is it okay? Is it visible? The entire screen?
Um, yes,
Is it visible? Okay. When we talk about nutrition, previously in the last decade, we used to talk about different foods taking and what are the health effects that the different foods have. But as time passed and as the genes, the genomic studies, the Human Genome Project completed in about 2000. There is a shift of the nutrition from the very traditional way of studying nutrition to a very deeply studying nutrition from the genetic aspects. With this Human Genome Project, they developed a number of areas of knowledge like genomics, proteomics, transcriptomics, metabolomics, and also foodomics. So all the different food items with their uh, with their biomolecules, with each of the different biomolecules that a food item is made of, has been studied, and their effects on the genes, the gene nutrient interaction, uh, that was studied, and we started a new domain of nutrition, you can say, that is personalized nutrition. uh, Similar to the personalized medicine, this new domain of personalized nutrition, or precision, or again, we can call it another by another term, that is precision nutrition. Mm, this which has got a very precise effect on the body, on the health, has started. And though it is in a very infant, or like a newborn baby, and it has not reached an adult stage, but the idea uh, is now available in most of the studies in the research form, but not in the clinical, in the clinical, in the hospitals, or uh, in the clinical research. It is available, but on the internships, or in the real arena, it is not so much available, because of the very costly methods of studying the gene and studying the biomarkers uh, of these nutrient gene interaction. About 2500 years ago, Hippocrates said that let food be thy medicine. Uh, in Ayurveda, ahar is a very important topic, and food, uh, and it was mainly the doctors, they prescribed foods that promote health in both disease and in normal people, and that is Mahavashya, that is called Mahavashya in Ayurveda. And this ahar has got is very much personalized ahar. That is, the population was divided into different categories according to their according to their needs, and according to their some specific uh, uh, characteristics, and based on those categorization, the Ayurvedic doctors prescribed the different food items for the different diseases, and for normal people also. Now, this is changed to, in the present form, we can say that let functional food be thy medicine. So what is this precision nutrition, or personalized nutrition, as you can again term it, is crucial, because it shifts from the general dietary recommendations. The general dietary recommendations are not absolute recommendations. They are constantly changing in uh, within 2 years or 3 years. The recommendations, the RDAs, that the ICMR is constantly changing, because uh, they are organizing different population based studies, and those on the based on those studies they are constantly changing the recommendations. And this shift towards personalized strategies, that are tailored for the individual's unique biological and environmental profile. So, this concept of personalized nutrition was first indicated by Hippocrates, according to whom your food is the base of your health. And according to Gallen, personal attitudes and unique responses to food, which are very important, and that led to this concept of precision nutrition. With the development of foodomics that I've already told about, that the tools that evolved during the Human Genome Project, in the last that started in last decade and ended in 2000, that led to that helped us, or that gave us those tools, that with those tools we can quantify or categorize the individual dietary intakes and their needs. So this is the concept of precision nutrition. And let us talk about what is about this foodomics. The foodomics is a term that is a very the from the omics technologies, when these technologies are used in terms of foods and in terms of nutrition, and that change the transcriptomics, the proteomics, the metabolomics. And what are these terms? That is the transcriptome, the entire array of transcriptomes, the entire group of proteins, the entire group of metabolites that are produced after the intake of a particular food, and the biostatistics and the chemometrics and the bioinformatics. So all of these technologies have helped us to understand the complex biological system that we have, and what are the mechanisms, how a bioactive food component, that is the bioactive component that is present in the functional food, how they interact with our genes, and how the genes are expressed. We all know that we have the all similar genetic makeup. Throughout the population, it is about 99 point about 6 or 7% same. So we are all same, but uh, but the expression of those genes, they are different. It is like a software and a hardware. The computers are all the hardwares where we use different types of softwares. The genes are the hardwares that we all have the same genetic makeup, but we are different in our functionality, in the production of the different proteins, or the product on, or how we digest, or our health, or our medicine, that the medicine that we take, and how the medicine acts against our genes, and how the vaccines that we take, they are controlled by the nutrition that we take, and that is the software. That is the software, that is the expression of the genes. That is very important, and that is different in each of us. So it is, the genes are same in all the people, but the expression of genes are different, and they are controlled by different factors in the environment, in the food, environment, exercise, and all these are the factors that controls the expression of the genes. There's a term called, I don't think it is there. Let me stick to these PPTs. So these are the foodomics. Here you can see the how the diet that affects the genes, and the genes the genes are expressed as mRNAs and the transcriptomes, and how the transcriptomes are translated into proteins, or the array of proteins, and how these proteins lead to the metabolomics, how these enzymes or proteins or enzymes that lead to the production of the metabolites. And here there is also the function of the gut microbiome. That is we are not only the made up of human cells. We are more than we are made up of microbial cells, and you can understand how because the number of microbial cell in our body is much higher than the human cells. So it is mainly controlled. So all the genetic expressions, they are controlled by these microbes and their metabolites, and how they produce those metabolites in our body. So here comes the factors that are involved in controlling all these processes. That is food safety, food traceability, food quality, and food bioactivity. So all these components, they are very much linked to the products that we produce in our body from food, and that help in controlling the different diseases and the normal condition of the body. All are dependent on these processes. This is a complete work of the foodomics strategies that includes very much analytical processes, because it requires high-end instruments, and so that they can evaluate the biomarkers. Like I've given an example in which a chicken egg spoilage after the treatment with 3 Pseudomonas bacteria commonly present in the matrices, that is the eggs inoculated with Pseudomonas aeruginosa, Pseudomonas fluorescens, and Pseudomonas putida, and their biomarkers, the old biomarkers were increase in lactic, succinic, and 3-hydroxybutyric acid. With the advent of all these UHPLC, high-end HPLC MSMS, new biomarkers have been found that decreases in 4 phospholipids, and that is phosphatidylcholine, lysophosphatidylethanolamine, and lysophosphatidylcholine. So all these are new biomarkers that are coming up, and that shows how the different bacteria, they act on the eggs, and they are present in these types of matrices. So personalized nutrition, the factors for personalized nutrition, what are the different factors? The number one is genetic variation. Though I told that it is the hardware is more or less the same in all the computers, but we have the generations. A computer, we always find that the hardwares, they are changing, and their cost is also changing. So hardware changes, and this genetic variations are sex, ethnicity, genetics, and environment. What is this environment? This environment is now termed as exposome, and this exposome, uh, that is the fetus exposed in the mother's womb, or within the mother's womb. They what are the molecules they are exposed to till death? Till death, from fetus to death, all the environment uh, takes an important or functions in a on the body, and this is called the exposome, the total effect of the environment, and that is important for the genetic variations. So sex, ethnicity, genetic, and environmental variations are important factors. Now, second is metabolism. What are the that I have already talked about? That is the microbes within our body. In the last year, not maybe the year before 2025, in 2024, another term came up, is aeronutrients. What is this aeronutrients? Aeronutrients, and another term is aeromicrobes. And what are these aeronutrients? We take about 9,000 liters of air every day, and with this air we take different types of molecules and different types of microbes also. The microbes constitute the aeromicrobes. When we go to a seaside, we take a lot of iodine along with this air, and so the people living in the seashore, they don't get the iodine deficiency disease, while people living in the mountains, they are suffering from iodine deficiency disease. So this availability of iodine, which is a very volatile component present in the seashore, air in the seashore, that helps to remove the deficiency in those people living in the seashore. And it is an example, a very good example of aeronutrients, and the vasculature within the lungs. It helps to take all these nutrients, and can help in combating the micronutrient deficiencies, because most of us are suffering from different diseases due to the micronutrient deficiencies. So this metabolic differences in metabolism is a very important part is the microbiota, or the microbial composition from the nasal tract, from the respiratory tract, the gastrointestinal tract, the urogenital tract, and all the tracts that are exposed to the environment. They are highly occupied by the microbes, and their and a variety, a diversity of microbes, and they are very diverse in nature, and they are helping. And also when they are there is a disbalance in their population, that cause a disease. So they are very important, and metabolomics depend on these microbes. Number 3 is absorption, because when we are taking the nutrients, we must absorb it. So it is also a very important part of personalized nutrition. The targeted dietary advice. The recommendations are based on extensive clinical and epidemiological traits, in which the generic cutoff points or levels are established to adequate nutrient intake for the population at large. So most of the recommendations, they without the recommendations, we cannot depend wholly on the personalized nutrition. We have to recommend some amount, because unless we do it, there will be a huge changes, or they will be very the intake, or the dietary intake will be in a mess, and that we can't do. So recommendations depend on the clinical and epidemiological studies, and they are very important. And we can't say that each people must get the personalized nutrition. We have to depend on those recommendations, or the RDAs. So what is then nutrigenomics? So nutrigenomics is a term that studies the interaction between the gene and the food. So it aims to understand how nutrition affects the genes and the expression of those genes, and vice versa. So nutritional genomics includes nutrigenomics, nutrigenetics, and nutritional epigenetics. So when we are talking about nutrition and its effect of genes, or the effect of genes on the nutrition, then we are talking about 3 important aspects. Now, number 1 is nutrigenomics. Number 2 is nutrigenetics, and the number 3 is epigenetics. So genes contains the instructions, or the hardwares, for how our body processes food. That is, it has that type. So this is a good example where gene nutrient interaction, how the gene nutrient interaction, there are 2 different types of foods here. The fats, mainly these types, these are the foods that gives minerals. These are the protein rich foods, and these are the vitamins. So what happens, what are the diseases the cardiovascular diseases, the how we enter into a, a format, or the disorders or the syndromes, and how what are the changes that takes place in the DNA, that is the genome. And that is mainly the methylation or the histone. There is histone modification, and these uh, DNA methylation leads to the different diseases. And there is a gene nutrient interaction, and these interactions play a crucial role in how nutrients are absorbed, are metabolized, are utilized by an organism, ultimately affecting growth, development, health, and disease susceptibility. It's very important about the way these gene nutrient interaction takes place within our body. So nutrigenomics concept, it's a omic concept, and nutrigenomics and nutrigenetics. And nutrigenomics, it includes also epigenetics. So nutrients and bioactive components of the foods, that leads to the different proteomics, that leads to the different types of metabolomics, the transcriptomics, from that these compounds have effect on transcriptomes, and ultimately there are differences in the phenotype, in the when it is in the changes in the genes, it is nutrigenetics. When it is changes in the RNA, it is epigenetics. The environment, the food, the exercise, these are the epigenetic factors that that changes the RNA, and that leads to the phenotype. These epigenetic factors, they can be controlled. We can reverse diabetes. We can do many things. We can do exercises to change the pattern of the diseases, and the methylation of the that takes place with different bad food items, we can say, but they can be reversed also. So epigenetics is such a component of this nutrigenomics that can reverse the process. So we have already seen in our COVID period that a person having COVID, he or she is absolutely fine, while a person living just on the same bed dies within 12 hours. So this happened due to a specific changes that specific or a different single nucleotide polymorphism, that is a specific change in the nucleotide that caused the change in the receptor of these COVID virus, the angiotensin converting enzyme in a receptor, that or the AC receptors, that has got about 10,000 single nucleotide polymorphism, and these single nucleotide polymorphism led to the affinity of the virus. And in the place where in a person where this affinity is very low, this the person 80% of the people, they didn't know that they have gone through COVID. but 20% of the people, and within that 20%, about 1% or 1.5, I didn't remember now, the specific group that died due to COVID. This is due to the high affinity or the SNPs that were found within that particular receptor of angiotensin converting enzyme. Similarly, all the diseases, they are due to this single nucleotide polymorphism on specific genes that have been identified after the Human Genome Project. The Human Genome Project is a very important, you can say that it is the it is mm, a scientific era which changed the entire knowledge uh, about us, and about ourselves, and about how the progression of the disease takes place, and why these diseases can't be reverted. And now we are going for specific gene editing technologies, and you can find it that CRISPR technologies, and there are some technologies that are coming up, and they are using those technologies. And we have also, you can find that within 1 to 2 months after the first COVID that came, we have developed the vaccine, whereas previously, for the vaccine to develop, it took about 10 years minimum. So, but for this time, the COVID, the exact structure of the vaccine that was developed in within 2 months. So this is possible due to these omics technologies, the bioinformatics, the nutrient-informatics, all the informatics technologies that came up, and that developed the structures. These are the different types of genes, and that caused the diseases. So Mexican people who consume a large portion of the carbohydrates and fats in their diet is associated with variants in the sweet taste receptors and cluster of differentiation C36, that is CD36. So they take a lot of carbohydrates and fats, and they are very much obese, and so because of their sweet receptor, that is the changes that have taken place in their sweet taste receptor. So these are some of the genes and their single nucleotide polymorphism that has changed, that has led to the disease. So omics-driven studies can be used for the development of these predictive models to determine the individual treatment strategies. So personalized nutrition is currently focused on people with metabolic disorders, with newborns, and with inborn errors of metabolism. There are about, I don't exactly know the number, but about 100 this inborn errors of metabolism has been identified, and these are some of the phenylketonuria, or these specific a change in a mutation in a particular gene led to these inborn errors of metabolism. These are very rare diseases, and the adults with monogenic disorders, hypercholesteremia, hypocatasia. So these are the monogenic disorders that are responsible for the for these diseases. So what's the difference between the nutrigenetics and the nutrigenomics? The 99.9%, as I have already told, that we are all same. Our hardware is same. So I have the same genes. So, but the variation is about 0.1%, and that makes the individual unique. That is the difference. That 1% is different. So then how this nutrigenetics, your genes affect how your body respond to food. The same food given to both the sister and the brother, they will have the different effect of the same food. And what is nutrigenomics? Nutrigenomics is the dietary component that changes the genetic expression of the same gene. So same gene present in 2%, but the same food having different gene expression. So that is nutrigenomics. So aspects, that is nutrigenetics, nutrigenomics. Nutrigenomics, study of how the nutrients and the dietary components influence gene expression, and the nutrigenetics is how the genetic variation, that is 1% variation, affect an individual's response to nutrients. The focus is diet to genes. The focus is genes to diet response. So level of action, gene expression in case of nutrigenomics. In the nutrigenetics, the DNA sequence variations, that is the single nucleotide polymorphisms. So they are very important in case of nutrigenetics. And the difference is due to those SNPs. The main question is, how does what we eat change the gene activity? And the main question for nutrigenetics is, why do people respond differently to the same diet? The outcome is identification of bioactive food components that modulate genes in case of nutrigenomics, while in case of nutrigenetics, identification of genetic variants that influence nutrient metabolism. So these are the differences between nutrigenomics and nutrigenetics. So application of nutrigenomics, development of functional food, dietary strategies to prevent disease, and for the nutrigenetics, the personalized nutrition based on genetic makeup. It is very important. So techniques used in case of nutrigenomics, the microarrays, RNA sequencing, epigenetic analysis. Like example, very good example is omega-3 fatty acids regulating inflammatory gene expression. During the COVID, we were engaged in making sprays with omega-3 fatty acids so that we can use inhalers as sprays for the omega-3 fatty acids that can reduce the inflammatory gene expressions. So broader population-based approach scope is, and for nutrigenetics, it is very much a personalized condition, like lactose intolerance. We can have that gluten-free diet for the people who develop allergies for gluten. So it is very much individual specific in case of nutrigenetics. So nutrigenetics and personalized diet. So single nucleotide polymorphism, epigenetic markers, copy number variations, and other genomic markers on biological responses to micronutrients and macronutrients. These are very important 4 points that are very important for personalized diets. Diet can directly impact the health outcome by influencing the expression of the genes involved in crucial metabolic pathways. It can also directly affect the health outcomes by influencing the frequency of the genetic mutations at the chromosomal or base sequence levels. Such in turn affects the expression of the genes. So diet can affect the gene expression, and the health effects of the nutrients depend on inherited genetic variants. How, what are inherited? We cannot only inherit our mother or father's that is knowledge. We must, or we inherit the genetic variations also, and that may lead us to disease, and so that alter the uptake and the metabolism of the nutrients and enzyme interactions at the molecular level and the nutrient cofactors, thereby affecting the biochemical reaction. So all these factors, the enzymes production, the microbes present in the gut, the nutrient cofactors that they are producing, that these microbes are producing, all these factors affect the nutrition. So this I can just skip this. I think transcriptomics is also a very strong. It needs. So this is metabolomics. That is very important, and that we can change also, because we can change the very nature of our microbes of the gut, and these microbes can change the metabolomics, that is 92 metabolic pathways and 400 metabolites in the humans for lung cancer biomarker discovery. So urinary biomarkers in particular can indicate the consumption of specific nutrients or food components. So these examples that include the menadione as a marker for vitamin K, hippuric acid as a marker for fruit and vegetable intake, 1-methyl histidine for oily fish, and methyl malonic acid for vitamin B12. So these are urinary marker. We can estimate or evaluate these markers, and we can say what are the needs of this of the patient, and how they can be changed with the diet, and how that can reduce the. So these are the lung cancer biomarkers, and they can change the metabolomics. So what is epigenetics? Epigenetics is something that I have already told about DNA methylation, how these transcriptomes and translations, these histones are changed according to the diet, and methyl groups are added to the DNA, and that can lead to the changes that can lead to disease, or that can lead to normal person. So histone modification is the main culprit that or the DNA methylation is the main culprit that is responsible for the different diseases. So this nutrients such as methionine, choline, folate, vitamin B12 are very important one carbon metabolism, and it is a pathway that produces methyl groups and necessary for the methylation of the DNA. So the deficiency or excess of these nutrients can disrupt normal epigenetic markers, leading to abnormal expression of genes and high risk of disease. So these deacetylation, methylation, phosphorylation, and other modifications. So these histone changes, the active state and the repressive state, these are the two states. One can lead to a disease, and the other can lead to lead to a disease due to abnormal expression of genes that can cause high risk of disease, and uh, the other can lead to that can help us to come up from the disease. So these are the bioactive components in the food. Some can lead to link to cancer, and some can be linked to CVDs. And these bioactive components on individual genotypes, the food contains many bioactive components, and there are polyphenols, flavonoids, saponins, polyunsaturated fatty acids that can cause single nucleotide that be responsible for changing this single nucleotide polymorphism, and in certain nucleotides in the genomes of different individuals, leading to different genotypes. And single nucleotide polymorphisms can lead to epigenetic changes and genetic variations that are linked to cancer and cardiovascular disease prevention. This is an example where fasting and food intake, what are the effects of these causes, the fasting or the food intake that changes the physiology of the body and the metabolism of the body, and there is epigenetic mechanisms and epigenetic changes. There is another very important topic that is in case of epigenetics, is that the epigenetic fat cell memory. When a person is in obese state, if you give a diet that for 3 months, for 6 months, and losing weight, that doesn't mean that when he or she will again move to the normal diet, he or she will retain that weight loss. This is due to the epigenetic fat cell memory of the adipose tissue, and it has been studied in humans and in mouse, and how the single nucleus RNA sequencing snRNAs, they were studied and they were compared, and they showed that due to the post weight loss relieved retention of obesity induced transcriptional changes in cells, and these mutations, these histone changes, the DNA methylations, they remain, and again the person gains weight, and again this epigenetic fat cell memory leads to again obesity. And it has been found that the bariatric surgery, in case of bariatric surgery, these people, it has been studied in European, most of the European countries who get very much this bariatric surgery, and there they found that after the surgery, within 1 or 2 months, they die, because this 20% or 25% loss of weight, that produce a lot of inflammatory markers are increased, and that cause heart attacks, and that produce that leads to death. So it's better to exercise normally, and it's not we can't just change our weight and think that it will remain because of this epigenetic fat cell memory. This is again a researcher from my group, who is now in Norway and working. She was in USA in this study. They found that how the alcohol induced, as we know that this Europe and America, USA, this alcohol induced DNA changes in pregnant mothers. They lead to children, and they develop alcohol syndrome, and this is due to the two genes, changes in the two genes within the womb, the POMC and the stress response, and the PER2 genes. These are the two genes that were due to the high level of alcohol during pregnancy. These genes change, and they give birth to babies that are not normal. So this shows the epigenetic changes. I think my time is nearly over. If you want, you can have some questions. I can have some questions.
Thank you very much, ma'am, for your insightful and enriching lecture.